Hello, everybody. In this lecture, we're going to conclude the different catabolic processes I want you to be familiar with. So let's get right into it. By the end of this module, you should be able to describe the roles of the pentose phosphate and interdeuteroff pathways, summarize the process of fermentation, describe different fermentation processes, and describe how lipids and proteins are broken down by extracellular and intracellular catabolism. So, glycolysis is evolutionarily conserved. Pretty much every cell has the ability to do glycolysis, whether it's prokaryotic or eukaryotic. However, there are some alternatives to the standard glycolytic pathway. These are still different. I think of them as different types of glycolysis, while some cells can have multiple forms of these glycolytic pathways. So there's this pentose phosphate pathway, and it's very similar to the regular glycolytic pathway, but what it can do is actually shift glucose away from the glycolysis pathway and move into this pentose pathway, which you can see down here, where we are going to actually end up with ribulose, and this can ultimately result in the formation of different nucleotides, or sorry, of nucleic acid sugars like pentose sugars like ribose and deoxyribose. And it also produces NADPH, which can be useful for the cell as well, because that can act as a coenzyme. Then there's the interdeuteroff pathway. Again, very similar to glucose for the normal glucose glycolysis pathway. This is thought to be an ancient pathway. It only produces 1 molecule of ATP per glucose, as opposed to the 2 molecules of ATP per glucose that we get from the regular glycolysis pathway, or the classical pathway. It also makes NADPH, which can later be used in different anabolic processes. And it's primarily found in gram-negative bacteria, specifically obligate aerobes, not found in humans. Pardon me. Now, the big thing I want you to know about these two pathways, the interdeuteroff and the pentose phosphate, is that they are alternatives to glycolysis that are going to start with your glucose molecule, which is going to get phosphorylated and ultimately end up in one of these pathways. All right. Now, if organisms don't have a terminal electron acceptor that can allow for a respiratory pathway, then they're going to actually transition into fermentation after they're done with glycolysis. And fermentation generally doesn't produce much or any ATP. What it does is it recharges your NADHs to NAD pluses. All right, so when you use glycolysis, 2 of your NAD pluses get converted to NADHs, and therefore they cannot be used in glycolysis again. What we need to do is convert them back to NAD pluses. They have to be, oxidized to NAD plus. And once they're oxidized back to NAD plus, they can re-enter the glycolytic pathway. So there are several different options of carbohydrate fermentation, lactic acid fermentation, alcohol fermentation, mixed acid, and butane diol fermentation. Excuse me. Now, in lactic acid fermentation, there are 2 pathways. There's the homolactic pathway, where the only thing that's produced is the lactic acid that we see down here on the left-hand side, right here. All right, lactate is the same as lactic acid. Fumarate is the same as fumaric acid. If you see that ATE, that's just the ion form of the acid. Pyruvate is pyruvic acid, etc. Now, in heterolactic fermentation, you get the formation of lactate and ethanol, as well as you can get carbon dioxide. You can get some other organic acids as well, like acetic acid and formic acid. In alcohol fermentation, this is what you get when you are, this is what yeast produce, right? When we're making beer or wine or something like that. And so you're going to go through the normal glycolysis pathway, and then pyruvate is going to be converted to ethanol with acetaldehyde as an intermediate. Now, the important part here is these NADHs, and this is really what's happening over here as well, but these NADHs are going to re-enter in the process of fermentation. You're going to get oxidized back to NAD pluses. They can then re-enter glycolysis. We've got butane diol, which is a offshoot here. We can see that over here we have acetate and ethanol being produced during fermentation. Pyruvate can also enter into this pathway where you form acetone and butane diol. We use a test called Voges Proscour to detect this in the lab. And you get some of the other organic acids, but you don't get them in very high quantities. You're mostly going to get the production of this butane diol, acetate, and ethanol. And then I skipped mixed acid. I did skip mixed acid. Sorry. So mixed acid and butane diol are part of the same like jam, more or less. You get a lot of the same stuff in butane diol that you get in mixed acid. You just get less of the organic acids. So you're gonna, the big identifier here is you get all these different organic acids: succinate, lactate, formate, and acetate. And you're going to produce CO2 and hydrogen ions as well, or hydrogen gas rather, as well. All right. Now, cells can also catabolize things other than sugars. Now, when they do that, usually it's going to break down outside of the cell to get to an intermediate product that'll be then brought into the cell. And so they use what are called exoenzymes or extracellular enzymes. They release those into the extracellular environment. Excuse me. And then that's going to break down those macromolecules or those polymers into smaller products. And things, these are enzymes, so their names mean something. Lipases break down lipids, proteases and peptidase break down proteins, nucleases break down nucleic acids. And so what you do is you then take these products, right? Carbohydrates go directly into glycolysis after they're broken down to simple sugars. Lipids enter here at the glyceraldehyde 3-phosphate. Fatty acids can be broken down to form acetyl-CoA, and then proteins can enter into the Krebs cycle at different points. And so this is kind of summarizing that. Your lipases are going to break your lipids into glycerols and fatty acids. And then the glycerol can be converted to DHAP, and DHAP is what enters into glycolysis. Fatty acids are broken down to form acetyl-CoA, which is the product of the intermediate step, which can then enter into the Krebs cycle. So when your proteins get broken down by your proteases and peptidases, they can be recycled to either make new proteins, right? So you could just reuse that amino acid, or they can be deaminated. And when you deaminate them, you release the NH2, and then they can enter into the Krebs cycle. And so here we show a couple of places where you are deaminating. In the process of deaminating, we lose ammonia. That's what the deamination means. Then you no longer have the ammonium over here, and this is going to be your deaminated product, which can then enter into the Krebs cycle. Nucleic acids like DNA and RNA aren't really metabolized a lot because they they often reuse those products because they're one of the main products that are going to contain phosphorus inside the cell. So if they can reuse them, that's oftentimes more energetically favorable. However, they can break down nucleic acids as well and enter into one of those various pathways for glycolysis or Krebs cycle. All right. So at this point, you should be able to tell me a little bit about what pentose phosphate and interdeuteroff are. Summarize the process of fermentation. So what is the main goal of fermentation? Describe how those different fermentation processes work, and how can lipids and proteins be broken down by extracellular and intracellular catabolism. So think about those different objectives and come to class with any questions that you might have about them. All right. Thank you very much. Talk to you all soon.